Microbial fermentation is a foundational technique in applied biology, harnessed to produce a wide range of economically and therapeutically important molecules, from small metabolites like ethanol, lactic acid, and antibiotics to complex macromolecules such as enzymes, polysaccharides, recombinant proteins, and vaccines. This process is based on the controlled growth of microorganisms under optimized environmental and nutritional conditions that channel their metabolic pathways toward the production of desired compounds. This process is based on the controlled growth of microorganisms under optimized environmental and nutritional conditions that channel their metabolic pathways toward the production of desired compounds. Fermentation can be classified broadly as either aerobic or anaerobic, depending on the oxygen requirement of the producing organism and the product in question. Fermenters are bioreactors of essential equipment used for industrial scale production, ranging from laboratory flasks to large stainless steel vessels with automated systems controlling pH, temperature, aeration, and agitation. In microbial fermentation, small molecules often refer to primary and secondary metabolites. Primary metabolites such as ethanol, citric acid, and amino acids, e.g., glutamic acid and lysine, are produced during the active growth phase of the organism and are essential for cellular functions. These are commonly produced by organisms like Saccharomyces cerevisiae, ethanol, Aspergillus niger, citric acid, and Corynebacterium glutamicum, amino acids. On the other hand, secondary metabolites like antibiotics, penicillin, streptomycin, erythromycin alkaloids, and pigments are synthesized in a stationary phase and are not directly involved in growth but often confer survival advantages. These are typically obtained from actinomycetes, streptomyces spp, fungi Penicillium spp, and certain bacteria. The production of macromolecules via fermentation involves more complex bioprocessing. Macromolecules include enzymes, e.g., proteases, amylases, cellulases, polysaccharides, e.g., xanthan gum, dextran, and recombinant proteins such as insulin, human growth hormone, and monoclonal antibodies. The production of recombinant macromolecules requires genetic manipulation of host cells like Escherichia coli, Saccharomyces cerevisiae, Pichia castorius, or mammalian cell lines using vectors containing appropriate promoters, regulatory sequences, and selection markers. Expression systems are chosen based on post-translational modification requirements of the product. For example, glycosylated proteins are better expressed in yeast or mammalian systems rather than in E. coli, which lacks the necessary machinery. Upstream processing and fermentation includes media preparation, inoculant development, and sterilization. The media composition is optimized for maximum yield of the desired product, often containing carbon and nitrogen sources, trace elements, and precursors. Downstream processing involves recovery and purification steps including cell disruption, if intracellular centrifugation, filtration, precipitation, chromatography, and drying. For high-value therapeutic proteins, purification must meet stringent regulatory standards to ensure safety, purity, and bioactivity. The yield and quality of fermentation products are influenced by parameters such as oxygen transfer rate, OTR, specific growth rate, biomass yield coefficient, and product formation kinetics, which are mathematically modeled using Monod and other fermentation kinetics models. Strain improvement is a critical aspect of microbial fermentation and includes random mutagenesis, adaptive laboratory evolution, and rational metabolic engineering. Recombinant DNA technology and CRISPR-based genome editing are increasingly used to engineer microorganisms for overproduction of target molecules, reduction of byproducts, and resistance to stress conditions such as high substrate or product concentrations. Systems biology approaches integrate omics data to model and optimize metabolic fluxes in industrial microbes. Microbial fermentation is also central to sustainable biotechnology, enabling production of bioplastics, e.g., polyhydroxyalkanoates or PHAs, biofuels, ethanol, butanol, biodiesel, and biodegradable chemicals from renewable biomass. Industrial microbiology increasingly relies on synthetic biology to design novel biosynthetic pathways and artificial regulatory circuits for the production of non-natural compounds. Moreover, continuous fermentation systems and immobilized cell technologies offer advantages in productivity, scalability, and cost-effectiveness. Fermentation-based bioprocesses have stringent regulatory oversight, particularly for pharmaceutical products. Agencies like the FDA and EMA require detailed characterization of the production organism, process validation, and rigorous quality control. Process analytical technology (PAT) is employed to monitor and control critical quality attributes in real time, ensuring consistency and compliance with good manufacturing practices (GMP). Previous year questions with answers. CSIR Net June 2018. Which of the following microorganisms is commonly used for the industrial production of glutamic acid? Escherichia coli. B. Streptococcus lactis. C. Corynebacterium glutamicum. D. Saccharomyces cerevisiae. Answer: C. Corynebacterium glutamicum. CSIR Net Deck 2016. Penicillin is produced as a secondary metabolite by which of the following organisms during fermentation? A. Aspergillus niger. B. Penicillium chrysogenum. C. Bacillus subtilis. D. Streptomyces griseus. Answer: B. Penicillium chrysogenum. CSIR Net June 2019. During recombinant protein production in E. coli, inclusion bodies are formed. What is the major reason? A. Improper folding due to lack of chaperones. B. Glycosylation of protein. C. High temperature and fermenter. D. Low expression of the protein. Answer: A. Improper folding due to lack of chaperones. Let me know the next topic whenever you're ready. You said. Application of immunological principles, vaccines, diagnostics. ChatGPT said. Here is your in-depth CSIR Net JRF Life Sciences lecture on applied biology, application of immunological principles, vaccines, diagnostics. Immunology forms the cornerstone of applied biology in both preventive and diagnostic medicine. The fundamental immunological principles such as antigen-antibody interactions, immune memory, clonal selection, and cytokine signaling have been exploited to develop effective vaccines and precise diagnostic tools. These applications are based on the ability of the immune system to distinguish self from non-self, mount a specific and memory-enhanced response against pathogens, and produce highly specific immunoglobulins that can be harnessed for both therapeutic and detection purposes. Vaccination is a classic example of applying immunological knowledge to elicit protective immunity. Vaccines work by introducing antigenic material into the body to stimulate the immune response without causing disease, leading to the generation of memory B and T cells. There are several types of vaccines based on the nature of the antigen used. Live attenuated vaccines, e.g., MMR, measles, mumps, rubella, contain weakened forms of the pathogen and typically induce strong cellular and humoral immunity with long-lasting protection. However, they may pose risks in immunocompromised individuals. Inactivated or killed vaccines, e.g., Salk polio vaccine, are safer but usually require booster doses. Subunit vaccines, e.g., hepatitis B, use purified antigens, often proteins or polysaccharides, and are produced via recombinant DNA technology. Toxoid vaccines contain inactivated bacterial toxins, e.g., tetanus, and stimulate the production of antitoxin antibodies. Conjugate vaccines combine weak antigens with strong carrier proteins to enhance immunogenicity, especially in children, e.g., Haemophilus influenzae type B vaccine. Recombinant vector vaccines and nucleic acid-based vaccines, DNA and mRNA, as in COVID-19 vaccines like Pfizer-BioNTech and Moderna, represent advanced vaccine technologies that rely on delivery of genetic material encoding pathogen-specific antigens. Adjuvants are key components of many vaccines, enhancing the immune response by stimulating innate immunity or increasing antigen uptake by antigen-presenting cells. Common adjuvants include aluminum salts, oil and water emulsions, and newer agents like toll-like receptor agonists. The development of vaccines involves rigorous preclinical and clinical trials, including immunogenicity testing, safety assessment, and efficacy evaluation through randomized controlled trials. Post-marketing surveillance is essential to monitor adverse events following immunization, AEFI. Diagnostics based on immunological principles utilize the specificity of antigen-antibody interactions. Enzyme-linked immunosorbent assay (ELISA) is a widely used technique for detecting antigens or antibodies in biological fluids, used for diagnosing infections, HIV, hepatitis, allergies, and autoimmune disorders. It relies on enzyme-labeled antibodies and a colorimetric or fluorometric readout. Western blotting, often used to confirm ELISA results, e.g., in HIV testing, separates proteins by electrophoresis and detects them using labeled antibodies. Lateral flow assays are rapid diagnostic tests, RDTs, or point-of-care devices like pregnancy tests or COVID-19 antigen tests, combining simplicity with specificity. Radioimmunoassay (RIA) is a highly sensitive technique for quantifying hormones, drugs, and viral antigens using radio-labeled antigens or antibodies. Immunofluorescence techniques use fluorescent-labeled antibodies to detect antigens in tissues or cells under a fluorescence microscope, commonly applied in detecting autoantibodies in systemic lupus erythematosus (SLE). Flow cytometry is a powerful diagnostic and research tool that quantifies and analyzes cell populations based on specific surface or intracellular markers, using fluorescently labeled antibodies. It is used extensively in immunophenotyping, leukemia diagnosis, and monitoring HIV progression, CD4+ T cell counts. Monoclonal antibodies (mAbs) are crucial in diagnostics and therapeutics. Produced via hybridoma technology, they are highly specific and reproducible. mAbs are used in immunoassays, targeted cancer therapies, e.g., rituximab for B-cell lymphoma, and blocking immune checkpoints in immunotherapy, e.g., PD-1 inhibitors. Advances in phage display, single B-cell sorting, and recombinant antibody engineering have led to the development of humanized and fully human antibodies to minimize immunogenicity in therapy. Immunodiagnostics also extend to detecting autoimmune diseases through the presence of specific autoantibodies, e.g., anti-nuclear antibodies in SLE, hypersensitivity reactions, e.g., skin prick tests for allergies, and monitoring immune function in transplant patients or immunodeficiency disorders. Newer multiplex platforms, such as bead-based assays and biosensors, allow simultaneous detection of multiple analytes, improving diagnostic throughput and accuracy. Recent advances include the integration of nanotechnology in immunodiagnostics and vaccine delivery, allowing targeted delivery, slow antigen release, and improved immunogenicity. mRNA vaccines, lipid nanoparticles, and self-amplifying RNA platforms are at the frontier of vaccine technology. Immune informatics and reverse vaccinology now allow in silico prediction of epitopes for designing subunit and peptide vaccines, bypassing traditional empirical methods. These innovations demonstrate how immunological principles are continuously evolving into next-generation applications. Previous year questions with answers. CSIR NetDeck 2017. Which of the following is not a characteristic feature of a live attenuated vaccine? A long-lasting immunity. B requires booster doses. C risk of reversion to virulence. D induces strong cellular and humoral response. Answer. B requires booster doses. Live attenuated vaccines typically provide long-lasting immunity with fewer boosters. CSIR Net June 2015. In an ELISA test for detection of antibodies, what did Answer. B requires booster doses. Live attenuated vaccines typically provide long-lasting immunity with fewer boosters. CSIR Net June 2015. In an ELISA test for detection of antibodies, what does the intensity of color developed indicate? A molecular weight of the antibody. B concentration of antigen. C affinity of antibody for antigen. D concentration of antibody in the sample. Answer. D concentration of antibody in the sample. CSIR Net Deck 2019. A patient has a chronic bacterial infection. Which diagnostic tool would best help in identifying the specific T-cell populations involved in the immune response? A Western blot. B ELISA C-flow cytometry. D RIA. Answer. C-flow cytometry. Let me know the next topic, and I'll continue building your perfect exam-focused lecture series. You said tissue and cell culture methods for plants. ChatGPT said. Here is your highly detailed CSIR Net JRF Life Sciences lecture on applied biology, tissue and cell culture methods for plants. Plant tissue and cell culture refers to the in vitro cultivation of plant cells, tissues, or organs under sterile, controlled environmental conditions on a nutrient medium. This technique is based on the principle of totipotency, the ability of a single plant cell to regenerate into an entire plant under the influence of suitable growth regulators and environmental cues. It has revolutionized plant biology, agriculture, and biotechnology by facilitating large-scale clonal propagation, germplasm conservation, genetic engineering, and production of valuable secondary metabolites. The starting material, or explant, can be any part of the plant, leaf, stem, root, shoot tip, node, or even a single cell. The explant is surface sterilized using disinfectants such as sodium hypochlorite, ethanol, or mercuric chloride to prevent microbial contamination. The sterilized explant is then inoculated onto a sterile nutrient medium such as Mershidi and Scoop MS medium, which contains essential macro and micronutrients, vitamins, carbohydrates, typically sucrose, and a gelling agent agar. The medium is supplemented with specific plant growth regulators, PGRs, auxins, e.g., 2, 4-D, Na, IAA, and cytokinins, e.g., BAP, kinidin, which govern the developmental pathway of the cultured cells. Callus culture is initiated when an explant is induced to form an unorganized mass of proliferating cells called a callus on a medium rich in auxins. Callus can be subcultured or used to initiate suspension cultures, where cells are grown in liquid medium under continuous agitation to produce uniform cell populations for secondary metabolite production. Suspension cultures are also used for protoplast isolation, somatic hybridization, and molecular studies. Organogenesis is the formation of organs like shoots and roots from the callus or directly from explants. A high cytokinin to auxin ratio favors shoot formation, while a high auxin to cytokinin ratio favors root formation. Alternatively, somatic embryogenesis refers to the development of embryo-like structures from somatic cells, which pass through the same stages as zygotic embryos: globular, heart-shaped, torpedo, cotyledonary, and can give rise to whole plants. This pathway is highly efficient for synthetic seed production and large-scale propagation. Micropropagation, or clonal propagation, involves the in vitro multiplication of elite, disease-free genotypes through axillary bud proliferation, adventitious shoot formation, or somatic embryogenesis. It is a key technology in commercial agriculture, horticulture, and forestry for producing genetically uniform and pathogen-free planting material. Stages of micropropagation include 1. Establishment, 2. Multiplication, 3. Rooting, and 4. Acclimatization, hardening in greenhouse conditions before transferring to soil. Anther and pollen culture enables the development of haploid plants through androgenesis. These haploids can be doubled to form homozygous diploids, doubled haploids, which are extremely valuable in plant breeding for rapid development of pure lines. Similarly, ovule and ovary culture can produce gynogenic haploids. Haploid production shortens breeding cycles and accelerates the development of stable hybrids. Protoplast culture involves the isolation of naked plant cells by enzymatic removal of cell walls using cellulase and pectinase. These protoplasts can be cultured to regenerate whole plants and are used for somatic hybridization, where protoplasts from different species are fused to create novel hybrids, somatic hybrids or hybrids. This allows gene transfer between sexually incompatible species, leading to creation of new cultivars with desirable traits such as disease resistance, stress tolerance, and high yield. Hairy root culture, induced by infection with agrobacterium rhizogenes, is used to produce high-value secondary metabolites and recombinant proteins. Hairy roots grow rapidly, are genetically stable, and can synthesize plant-specific products at high yields, such as alkaloids and flavonoids. In recent years, cell suspension cultures have been optimized in bioreactors for commercial production of secondary metabolites like chiconin, paclitaxel, taxol, and berberine. Cryopreservation is used for long-term storage of elite germplasm and transgenic lines. Moreover, synthetic seed technology involves encapsulating somatic embryos in a gel matrix to mimic true seeds, facilitating easy storage and transportation. Plant tissue culture also plays a critical role in genetic transformation, especially using agrobacterium tumefaciens-mediated transformation or vialistic methods. After gene transfer, transformed cells are selected using antibiotic resistance markers and regenerated into whole plants through tissue culture methods. Previous year questions with answers. CSIR Net June 2016. In plant tissue culture, a high cytokinin to auxin ratio in the medium generally promotes a callus formation. B root formation, C shoot formation, D embryo formation. Answer: C shoot formation. CSIR Net Deck 2018. Which of the following is a correct application of protoplast fusion in plant biotechnology? A haploid production. B development of cytoplasmic hybrids, hybrids. C acclimatization of micropropagated plants. D root induction in tissue culture. Answer: B development of cytoplasmic hybrids, hybrids. CSIR Net June 2015. Which plant tissue culture method is commonly used to generate virus-free plants? A callus culture. B suspension culture, C meristem culture, D anther culture. Answer: C meristem culture. Let me know the next topic whenever you're ready. You said, and animals. ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on applied biology, tissue and cell culture methods for animals, highly detailed, JRF level, paragraph-based, exam-oriented. Animal cell and tissue culture is a foundational technique in modern biology that involves the maintenance and propagation of animal cells, tissues, or organs under controlled laboratory conditions. It serves as a powerful platform for physiological, biochemical, genetic, and pharmacological studies and is widely used in vaccine development, cancer biology, regenerative medicine, and production of therapeutic proteins. The technique requires sterile environments, specialized culture media, and incubators that maintain optimal temperature, pH, oxygen, and CO conditions. There are two main types of animal culture systems: primary cultures and cell lines. Primary cultures are derived directly from animal tissues by mechanical or enzymatic disaggregation using enzymes such as trypsin or collagenase. These cells closely resemble the original tissue both morphologically and functionally but have limited lifespan and undergo replicative senescence after a few passages. If primary cultures undergo transformation, spontaneous or induced, they can become immortalized cell lines, capable of indefinite proliferation. Immortalization can occur naturally, as in cancer cells, or be induced by viral oncogenes, e.g., SV40T antigen telomerase activation, or carcinogens. Cell lines are classified as finite or continuous. Finite cell lines have limited doubling capacity and show contact inhibition, whereas continuous cell lines such as HeLa, human cervical cancer cells, CHO, Chinese hamster ovary, and HEK293, human embryonic kidney, are immortal and widely used in research and industry. Adherent cells require a solid substrate for growth, e.g., fibroblasts, whereas suspension cells like lymphocytes can grow freely in culture medium. The culture medium must supply essential nutrients, vitamins, salts, amino acids, glucose, and growth factors. Commonly used basal media include Eagle's minimum essential medium, MEM RPMI 1640, and DMEM Delbeco's modified Eagle medium. These media are often supplemented with fetal bovine serum, FBS, which provides hormones and attachment factors. Serum-free media have been developed for applications requiring defined conditions or to avoid animal-derived components. The most commonly used animal cell culture techniques include monolayer culture, suspension culture, organ culture, and 3D culture systems. Organ culture maintains tissue architecture and functionality and is useful for developmental and toxicological studies. In contrast, 3D culture systems, e.g., spheroids, organoids, scaffold-based cultures, provide a more physiologically relevant microenvironment for studying cell and cell matrix interactions, especially in cancer and stem cell research. Cryopreservation is routinely used to store cell lines at ultra-low temperatures, usually in liquid nitrogen at -196 degrees Celsius, using cryoprotectants like DMSO or glycerol to prevent ice crystal formation. This ensures the long-term availability of cell stocks and minimizes genetic drift or contamination due to repeated passaging. A major application of animal cell culture is in biopharmaceutical production. Mammalian cells, especially CHO cells, are used to produce therapeutic proteins such as monoclonal antibodies, insulin analogs, and interferons with human-compatible post-translational modifications. Hybridoma technology, developed by Kohler and Milstein, combines antibody-producing B cells with myeloma cells to produce monoclonal antibodies, which are indispensable in diagnostics, therapeutics, and research. Vaccine production is another key application. Viral vaccines such as those against polio, rabies, influenza, and hepatitis B are produced in cultured cell lines like Vero, BHK21, and MDCK. Cell cultures provide a safe, reproducible, and scalable platform for mass vaccine manufacturing. Recent advances include use of transient expression systems in HEK293 or CHO cells for rapid production of subunit or recombinant vaccines, as demonstrated in COVID-19 vaccine development. Stem cell culture is central to regenerative medicine and developmental biology. Embryonic stem cells, ESCs, are pluripotent and can differentiate into all three germ layers. Adult stem cells, such as mesenchymal stem cells, MSCs, and hematopoietic stem cells, HSCs, have multipotent potential and are used in therapies for degenerative diseases, hematological disorders, and tissue repair. iPSCs, induced pluripotent stem cells, are generated by reprogramming somatic cells with transcription factors October 4, SOX2, KLF4, CMYC, offering patient-specific therapeutic options. Another frontier in animal cell culture is gene editing and transgenesis. Cultured cells are manipulated using CRISPR, SOS9, TALENs, or ZFNs to study gene function or produce genetically modified animals. Transfection methods include chemical, liquefaction, physical, electroporation, and viral lentiviral vectors. Stably transfected cells are selected using markers like antibiotic resistance and clone for further use. Quality control is vital in animal cell culture to avoid issues like mycoplasma contamination, which is common, difficult to detect, and can alter cellular metabolism and experimental outcomes. Cells must be periodically authenticated using DNA fingerprinting, karyotyping, and STR profiling. In summary, animal tissue and cell culture is an indispensable tool in modern life sciences, enabling detailed analysis of cell behavior, drug discovery, vaccine production, stem cell therapy, and molecular medicine. The continuous refinement of these techniques is driving the development of precision and personalized medicine. Previous year questions with answers. CSIR Net Deck 2016. Which of the following is the most appropriate method to generate monoclonal antibodies? A somatic hybridization between B lymphocytes and myeloma cells, B fusion of two myeloma cell lines, C transformation of B cells with viruses, D in vitro maturation of B cells. Answer: a somatic hybridization between B lymphocytes and myeloma cells. CSIR Net June 2017. In animal cell culture, why is DMSO added during cryopreservation? A to provide nutrients. B to increase oxygen concentration, C to prevent ice crystal formation, D to induce transformation. Answer: C to prevent ice crystal formation. CSIR Net Deck 2019. Which of the following cell lines is most commonly used for large-scale monoclonal antibody production? A. HeLa. B. NIH3T3 CHO DHEK293 Answer: CHO. Let me know the next topic, and I'll continue your lecture series at the same standard. You said, transgenic animals. ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on applied biology, transgenic animals. Extremely detailed, paragraph-based, CSIR Net JRF standard, with maximum word coverage and previous year questions. Transgenic animals are genetically modified organisms, GMOs, in which foreign genes, transgenes, are inserted into their genome to study gene function, model diseases, or produce valuable biological products. These animals carry and express exogenous genes introduced through artificial genetic manipulation, making them indispensable in functional genomics, biomedical research, and biotechnology. The process of generating transgenic animals involves the stable integration of the desired gene into the host genome in a manner that ensures its expression across tissues or specific developmental stages. The most commonly used transgenic animals are mice, due to their small size, rapid breeding cycle, well-characterized genome, and close genetic similarity to humans. Other animals like rats, rabbits, sheep, pigs, and cows are also used depending on the purpose, such as disease modeling, pharmaceutical production, or agricultural enhancement. Methods of transgenesis include pronuclear microinjection. This is the most traditional and widely used technique in which a DNA construct containing the gene of interest is microinjected into the male pronucleus of a fertilized zygote. The zygote is then implanted into a pseudopregnant female. This method is random and may result in variable gene expression due to position effects or gene silencing. Embryonic stem S cell-mediated gene transfer. Here, S cells derived from the inner cell mass of a blastocyst are genetically modified in vitro and then injected into a recipient blastocyst to generate chimeric animals. If the S cells contribute to the germline, the transgene can be passed to the next generation. This method allows targeted gene modification, including gene knockouts and knock-ins via homologous recombination. Retroviral or lentiviral vectors. These viruses are used to deliver transgenes into early-stage embryos or zygotes. Retroviruses integrate into the host genome, ensuring stable inheritance. Lentiviruses can infect both dividing and non-dividing cells and are highly efficient, although they may have size limitations for the transgene. CRISPR, SOS9-mediated genome editing. This revolutionary technique enables precise and efficient gene editing at specific loci. CRISPR allows for generation of gene knockouts, knock-ins, point mutations, conditional alleles, and large genomic rearrangements. When applied to embryos, CRISPR can produce transgenic or genome-edited animals in a single generation without requiring S cells or viral vectors. Applications of transgenic animals. Functional genomics and gene function studies. Transgenic mice expressing or lacking specific genes help in understanding gene function, signaling pathways, and developmental biology. Knockout mice are invaluable for determining gene essentiality, while knock-in mice allow study of disease-causing mutations. Disease models. Transgenic animals replicate human diseases, including cancer, diabetes, neurodegenerative diseases like Alzheimer's and Parkinson's, and cardiovascular disorders. For example, APS1 transgenic mice express human mutant genes linked to Alzheimer's, making them a gold standard for drug testing. Pharmaceutical production. Farming. Transgenic livestock have been engineered to express therapeutic proteins like clotting factors, growth hormones, antibodies, and enzymes in their milk, eggs, or blood. An example is Atrin, an anticoagulant produced from the milk of transgenic goats expressing human antithrombin-3. Xenotransplantation. Transgenic pigs with human-compatible antigens are being developed as potential organ donors to address the shortage of human organs for transplantation. Genetic modification reduces immunological rejection by eliminating alpha-1, free galactosyl-transferase gal-epitope and incorporating human complement regulatory proteins. Agricultural improvements. Transgenic animals with enhanced traits such as faster growth, disease resistance, and improved feed conversion efficiency are being developed. The EnviroPig, for instance, expresses phytase in its saliva, which allows it to digest phosphorus more efficiently, reducing environmental pollution. Biosafety and environmental research. Transgenic fish and insects are used to study ecological interactions, monitor pollutants, and reduce vector-borne diseases. Genetically engineered sterile male mosquitoes, for instance, are released to suppress wild populations and control diseases like dengue and malaria. Despite these benefits, transgenic animal research raises several ethical, ecological, and biosafety concerns. Ethical issues involve animal welfare, pain, suffering, and unintended health effects due to insertional mutagenesis. Biosafety concerns include gene flow to wild populations and unintended ecological impacts. To address these, stringent regulations are enforced by organizations such as CPCSEA, India, NIH, USA, and OECD. In India, transgenic animal research is regulated by DBT and PEAK, and all protocols require approval under guidelines such as the rules for manufacture, use, import, export, and storage of hazardous microorganisms (GMOs) under the EPA 1986. Animal ethics and humane handling are overseen by the Institutional Animal Ethics Committee (IAEC) and CPCSEA. The transgene construct typically includes a gene of interest (GOI), a promoter like CMV, or tissue-specific ones like beta-casein promoter for mammary gland expression, enhancers, and selection markers. Transgene expression may vary based on position effect, copy number, epigenetic silencing, and integration site. Techniques like Southern blotting, PCR, qPCR, and Western blotting are used for transgene detection and expression analysis. In conclusion, transgenic animals represent a transformative approach to studying gene function, modeling human diseases, and producing biopharmaceuticals. With evolving gene editing technologies like CRISPR, the efficiency, specificity, and applications of transgenesis are rapidly expanding, opening new frontiers in personalized medicine, regenerative therapies, and sustainable agriculture. Previous year questions with answers. CSIR-Net June 2015. Which of the following methods is best suited for creating gene knock in animals? A pronuclear microinjection. B. Retroviral transduction CCR-ISPR SOS9 with homologous recombination D. Somatic cell nuclear transfer answer. CCR-ISPR SOS9 with homologous recombination. CSIR-Net Deck 2018. Transgenic goats are used to produce which of the following human therapeutic proteins? A. Erythropoietin. B. Human growth hormone C. Antithrombin 3 D. Insulin. Answer. C. Antithrombin 3. CSIR-Net June 2020. Which of the following is not an advantage of CRISPR SOS9 over traditional gene knockout techniques in animals? A faster generation of mutants. B. High off-target effects. See no need for embryonic stem cells. See applicable to many species. Answer. B. High off-target effects is a disadvantage, not an advantage. Let me know your next topic. I'll continue with the same level of quality and detail. You said, and plants, ChatGPT said. Here is your CSIR-Net JRF Life Sciences lecture on applied biology, transgenic plants. Maximum word limit, paragraph style, no tables, JRF level notes based strictly on CSIR-Net syllabus and previous year questions. Transgenic plants are genetically modified plants that contain one or more genes artificially inserted using biotechnological methods. These genes may come from unrelated species, including bacteria, viruses, or even animals. The inserted gene, known as a transgene, integrates into the plant's genome and is stably inherited by subsequent generations. The purpose of developing transgenic plants includes enhancing crop yield, improving resistance to pests, diseases, and abiotic stress, enhancing nutritional value, and producing pharmaceuticals or industrially relevant proteins, also called molecular farming. The development of transgenic plants begins with identifying a gene of interest and cloning it into a plant transformation vector, which typically contains a promoter, commonly the CAMB35S promoter terminator sequences, selectable markers such as antibiotic resistance genes like NPTII or herbicide resistance genes like BAR, and reporter genes like GUS or GFP. This construct is then introduced into plant cells using transformation methods. The transformed cells are selected and regenerated into whole plants using plant tissue culture techniques. Major plant transformation methods include agrobacterium-mediated transformation. This is the most widely used method, particularly for dicots. Agrobacterium tumefaciens naturally transfers part of its T plasmid DNA, tDNA, into the plant genome during infection. Scientists exploit this ability by replacing the tumor-inducing genes in tDNA with genes of interest. The modified agrobacterium is co-cultivated with plant explants like leaf discs, and after successful integration, the cells are regenerated into whole plants using growth hormones. Biolistics particle bombardment, especially useful for monocots like rice, wheat, and maize which are less susceptible to agrobacterium. In this method, DNA-coated gold or tungsten particles are physically shot into plant tissues or cells using a gene gun. The DNA integrates randomly into the plant genome. Though less precise, this method is highly effective for species recalcitrant to agrobacterium. PEG-mediated electroporation. These are used for protoplast transformation. Polyethylene glycol (PEG) or electrical pulses allow the DNA to enter protoplasts, plant cells without cell walls, which are then regenerated. However, the regeneration of whole plants from protoplasts can be challenging in many species. Once transformed, cells are selected using antibiotic or herbicide resistance and induced to regenerate through organogenesis or somatic embryogenesis using tissue culture techniques. Transgenic plants are screened using molecular techniques like PCR, Southern blotting, RT-PCR, and Western blotting to confirm transgene presence and expression. Applications of transgenic plants. Insect and pest resistance. Transgenic crops expressing Bacillus thuringiensis (BT) toxin genes like Cry1AC have revolutionized pest management. BT cotton, for example, produces a protein toxic to bollworms but safe for humans and non-target organisms. This reduces pesticide use and enhances yield. Herbicide tolerance. Crops like soybean and canola have been engineered to tolerate broad-spectrum herbicides such as glyphosate, e.g., Roundup-ready crops. This allows farmers to apply herbicides for weed control without damaging the crop, improving farm efficiency. Disease resistance. Transgenic plants can carry viral coat protein genes or RNA interference (RNAi) constructs to confer resistance against viruses, bacteria, or fungi. An example is papaya ring spot virus-resistant papaya, which saved the Hawaiian papaya industry. Abiotic stress tolerance. Genes for osmoprotectants, ion transporters, or transcription factors e.g., DREB, HPA have been used to create drought, salt, and cold-tolerant plants, especially important under changing climate conditions. Nutritional enhancement biofortification. The most famous example is golden rice, engineered to produce protein A, beta-carotene in the endosperm. Transgenic rice, banana, and cassava are also being developed to address vitamin and mineral deficiencies in developing countries. Pharmaceutical production. Molecular farming. Plants like tobacco and maize are engineered to produce antibodies, vaccines, and therapeutic proteins. This low-cost, scalable system is being developed for oral vaccines and biologics like insulin or hepatitis B surface antigen. Phytoremediation. Transgenic plants expressing genes for metal transporters or degradative enzymes can be used to remove pollutants like heavy metals, petroleum hydrocarbons, and explosives from contaminated environments. Despite these benefits, transgenic plants are under strict biosafety regulation due to concerns over gene flow to wild relatives, development of resistant pests, ecological imbalance, and food safety. In India, regulatory oversight is provided by GEEC Genetic Engineering Appraisal Committee and RCGM Review Committee on Genetic Manipulation under DBT, as per the EPA 1986 rules. Gene containment strategies such as male sterility, chloroplast transformation, which avoids pollen-mediated gene flow, and terminator technology go controversial or under exploration to address biosafety concerns. From a scientific perspective, the success of a transgene depends on factors like copy number, insertion site, position effect, gene silencing, and epigenetic regulation. Modern technologies like RNA interference (RNAi) and CRISPR (SOS9) now offer more precise gene regulation and editing, enabling development of cisgenic from the same species or intergenic plants, which may reduce regulatory concerns. In conclusion, transgenic plants offer transformative solutions for sustainable agriculture, food security, environmental protection, and global health. With precise genetic tools and robust regulatory frameworks, their safe and effective use can greatly enhance human welfare and ecosystem resilience. Previous year CSIR-Net questions with answers. CSIR-Net Deck 2014. Golden rice is a transgenic crop engineered to produce iron. B vitamin A, beta carotene, C vitamin C. D folate. Answer: B vitamin A, beta carotene. CSIR net June 2016. In a transgenic plant expressing Cry1AC gene from Bacillus thuringiensis, the protein product is effective against a bacterial blight. B fungal rust C. Lepidopteran larva D. aphids. Answer: C. Lepidopteran larva. CSIR net Deck 2019. Which of the following techniques ensures minimal gene flow from transgenic plants to wild relatives? A. Using antibiotic resistance markers. B. Chloroplast transformation. C. Biolistic transformation. D. Using herbicide resistance genes. Answer: B. Chloroplast transformation. Let me know your next topic. I'll deliver it with the same CSIR JRF level depth and precision. You said, and plants, ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on applied biology, transgenic plants. JRF level, maximum word limit, paragraph format, high quality, based on previous year questions and syllabus emphasis. Transgenic plants are genetically modified organisms, GMOs, in which foreign DNA has been artificially introduced into the plant genome using recombinant DNA technology. This modification allows plants to express desirable traits that may not naturally exist within the species, such as pest resistance, herbicide tolerance, improved nutritional quality, or production of pharmaceutical proteins. The concept is rooted in the ability to stably integrate and express an exogenous gene, transgene, in plant cells that can be regenerated into whole, fertile plants through tissue culture techniques. The development of a transgenic plant begins with the identification and isolation of the gene of interest. This gene is then inserted into a plant transformation vector, often derived from a modified T tumor-inducing plasmid of Agrobacterium tumefaciens or into a plasmid used in biolistic methods, gene gun. The gene is typically placed under the control of a strong, constitutive plant promoter, e.g., CAMB35S, along with a selectable marker gene such as NPTII, conferring kanamycin resistance or BAR conferring resistance to the herbicide phosphenoprisin. After vector construction, the DNA is introduced into plant cells through agrobacterium-mediated transformation, biolistics, electroporation, or PEG-mediated transformation of protoplasts. Agrobacterium-mediated transformation is the most widely used method, particularly effective in dicots and some monocots. The bacterium naturally transfers a segment of its T plasmid, tDNA, into the host plant genome. In transgenic protocols, the oncogenic genes in tDNA are replaced with the gene of interest. The bacterium infects wounded plant tissue, like leaf discs, and the transformed cells are selected on a medium containing a selective agent. These cells are then regenerated into whole plants using hormones in the culture medium, a process relying on somatic embryogenesis or organogenesis. Particle bombardment gene gun method is used for plants recalcitrant to agrobacterium, especially cereals like maize, rice, and wheat. DNA-coded metal particles, gold or tungsten, are accelerated into plant tissues. Though efficient, this method may result in random insertion, multiple copies of transgenes, and gene silencing due to positional effects or rearrangements. Once transformed, confirmation of integration is done using PCR, Southern blot, RT-PCR, and Western blot, ensuring that the transgene is present, transcribed, and translated. Gene expression can be controlled using tissue-specific promoters or inducible systems. Transgenic plants are also screened for unintended effects and phenotypic stability across generations. Applications of transgenic plants are diverse. Insect resistance. Expression of cry genes from Bacillus thuringiensis (BT) leads to production of delta endotoxins that kill Lepidopteran larvae. BT cotton, the first transgenic crop in India, drastically reduced pesticide use and increased yield. BT brinjal is another example engineered against fruit and shoot borer, though its commercial release is debated. Herbicide tolerance. Transgenic crops like Roundup Ready soybean and Liberty Link maize express genes such as CP4 enabling survival under glyphosate or glufosinate treatment. This allows broad-spectrum weed control with minimal damage to the crop. Disease resistance. Viral coat protein-mediated resistance and RNA interference (RNAi) have been used in developing plants resistant to viruses such as papaya ring spot virus and tomato yellow leaf curl virus. RNAi has also been used to silence genes in pathogens or pests, a method known as host-induced gene silencing pigs. Abiotic stress tolerance. Plants expressing transcription factors like DREB, CBF, or HbA1 show improved resistance to drought, salinity, or cold by modulating stress-responsive genes. Overexpression of genes for osmoprotectants like proline or glycine betaine also confers tolerance. Nutritional enhancement. Golden rice, a milestone in biofortification, expresses Cy and CRT genes from maize and bacteria respectively to produce beta-carotene in the endosperm. Similarly, transgenic bananas, cassava, and maize are being engineered to enhance iron, zinc, and essential amino acids. Pharmaceutical production. Plantibodes. Plants like tobacco and alfalfa are used to produce antibodies, vaccines, and therapeutic proteins, such as hepatitis B surface antigen or insulin. This is part of molecular farming, a cost-effective alternative to microbial or animal-based systems. Phytoremediation. Transgenic plants expressing metal-binding proteins or degradative enzymes are used to clean up environments contaminated with heavy metals or explosives like TNT. Arabidopsis and brassica species have been genetically modified for this purpose. Industrial enzyme production. Plants have been engineered to produce enzymes like phytase to improve phosphate absorption in animals or cellulases for biofuel production. To prevent environmental risks such as gene flow, several containment strategies have been proposed, including chloroplast transformation, which is maternally inherited in most plants and avoids pollen-mediated gene escape, male sterility, and terminator technology, genetic use restriction technology, GURT, though the latter faces ethical and legal resistance. Regulatory bodies such as the Genetic Engineering Appraisal Committee in India evaluate transgenic crops for environmental and food safety under the rules of 1989 of the Environment Protection Act, 1986. Only after rigorous biosafety assessments are transgenic crops approved for commercial release. Transgenic research is also monitored by RCGM, Review Committee on Genetic Manipulation, under DBT. With advancements in genome editing tools like CRISPR-Cas9, newer generations of transgenic plants, often referred to as genome-edited resistant plants, are being developed with precise genetic modifications and minimal off-target effects. These methods also face lesser regulatory resistance compared to traditional GMOs in some countries. In summary, transgenic plants have the potential to revolutionize agriculture by providing sustainable solutions to food security, malnutrition, environmental pollution, and industrial challenges. Their development demands rigorous scientific methodology, robust regulatory oversight, and transparent public dialogue. Previous year CSIR net questions with answers. CSIR net Deck 2014. Golden rice is a transgenic crop engineered to produce a vitamin C. B vitamin A, beta carotene, C iron. D zinc. Answer: B vitamin A, beta carotene. CSIR net June 2016. Which of the following cry genes is most effective against Lepidopteran insects? A cry1AC. B cry3b flat, C cry9, D cry2a flat. Answer: a cry1AC. CSIR net Deck 2019. Which method of gene transfer is least likely to lead to pollen-mediated gene flow? A agrobacterium-mediated transformation. B biolistics. C chloroplast transformation. D protoplast fusion. Answer: C chloroplast transformation. Let me know your next topic. I'll prepare the next lecture with the same JRF level focus and quality. You said. Molecular approaches to diagnosis and strain identification. ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on applied biology, molecular approaches to diagnosis and strain identification, maximum word limit, JRF level, previous year questions with answers included, no tables, full paragraph format. Molecular approaches to diagnosis and strain identification are central to modern biology, medicine, agriculture, and environmental microbiology. These methods rely on the analysis of nucleic acids, DNA, and RNA proteins, or metabolites to precisely detect, identify, and differentiate organisms at the species, strain, or even single nucleotide level. Molecular techniques are favored over classical phenotypic methods due to their higher sensitivity, specificity, reproducibility, and the ability to detect unculturable or rare organisms. The fundamental basis of molecular diagnosis involves the detection of unique genetic signatures, such as species-specific or strain-specific DNA sequences, using nucleic acid amplification, hybridization, or sequencing technologies. The target may be a pathogen, genetic marker, or mutation, and the sample can be derived from clinical material, blood, tissue, saliva, environmental sources, soil, water, or agricultural products, leaves, seeds. The most widely used molecular diagnostic tool is the polymerase chain reaction (PCR). PCR amplifies specific DNA sequences exponentially using sequence-specific primers, a thermostable DNA polymerase like Taq DNTPs, and thermal cycling. Conventional PCR is qualitative, detecting the presence or absence of a target. However, for diagnostic accuracy, quantitative real-time PCR (qPCR) is preferred. It uses fluorescent probes or interpolating dyes to monitor amplification in real time, allowing quantification of the target. This is crucial for viral load determination, e.g., HIV, HPV pathogen burden, and early detection. Reverse transcription PCR (RT-PCR) is used when RNA is a template, such as in detection of RNA viruses like SARS-CoV-2 or dengue virus. Another highly specific method is DNA hybridization, which uses labeled DNA or RNA probes to bind complementary sequences. This is the basis for dot blot, southern blot, and in situ hybridization. Though labor-intensive, these methods are useful for strain typing, especially in combination with restriction enzyme digestion patterns, e.g., RFLP, restriction fragment length polymorphism. In RFLP, genomic DNA is digested, and the variation in banding pattern upon gel electrophoresis reflects polymorphisms useful for strain identification in bacteria, fungi, or plants. Multiplex PCR is another advanced diagnostic tool, allowing simultaneous amplification of multiple targets in a single reaction. It is often used in detecting co-infections or in pathogen panels for differential diagnosis. Nested PCR, involving 2 rounds of amplification with internal primers, is used to enhance specificity and sensitivity, especially in samples with low DNA content. Molecular strain typing is essential in epidemiology, particularly for tracking outbreaks and determining the source of infections. Techniques such as pulse field gel electrophoresis (PFGE) resolve large DNA fragments, creating unique fingerprints for bacterial strains. PFGE was a gold standard for many years in identifying clonal relationships among Salmonella, Listeria, or E. coli strains. Random amplified polymorphic DNA (RAPD) and amplified fragment length polymorphism (AFLP) are PCR-based techniques that generate strain-specific banding patterns without prior sequence knowledge. These are useful in strain typing of fungi, plants, and microbial isolates from natural environments. With the advent of next-generation sequencing, NGS, strain identification has reached unparalleled resolution. Whole genome sequencing (WGS) can identify single nucleotide polymorphisms, SNPs, insertions, deletions, and genome rearrangements, providing complete genomic blueprints for comparison. This is the most powerful tool in molecular epidemiology, allowing accurate identification of pathogenic strains, antibiotic resistance genes, and virulence factors. Metagenomic sequencing allows diagnosis directly from environmental or clinical samples, bypassing the need for culturing. In clinical diagnosis, nucleic acid-based diagnostics have revolutionized pathogen detection. Commercial platforms like GenExpert, FilmArray, or LAMP loop-mediated isothermal amplification offer rapid, point-of-care testing. LAMP uses strand-displacing DNA polymerases in multiple primers, producing visible results in less than an hour without thermal cycling, making it highly suitable for field diagnostics, especially in resource-limited settings. Another key area is molecular barcoding, where conserved genes like 16S rRNA in bacteria, ITS, fungi, or COI, cytochrome oxidase, and in animals are sequenced for identification. These gene regions are chosen for their variability between species but conservation within species, allowing accurate taxonomic identification. This is widely used in microbiome studies and biodiversity surveys. Microarrays are another powerful diagnostic tool, consisting of thousands of immobilized oligonucleotide probes on a chip. They can simultaneously detect hundreds of pathogens or mutations in a single assay. SNP arrays are used in genotyping and strain differentiation in plants, animals, and microbes. In the agricultural and veterinary domains, molecular diagnosis is crucial for early detection of pathogens causing crop or animal diseases. For instance, PCR-based kits are used for phytophthora infestans in potatoes, xanthomonas in rice, and foot-and-mouth disease virus (FMDV) in livestock. Molecular methods are also used to confirm GMO status, detect mycotoxigenic strains of fungi, and differentiate biocontrol strains from pathogens. Molecular identification is indispensable in biotechnological applications such as strain validation and industrial fermentations, e.g., confirming recombinant E. coli or Saccharomyces cerevisiae, tracking contaminating organisms, and ensuring clonal purity. Finally, biosensors combining molecular recognition elements, DNA, antibodies with signal transducers, optical, electrochemical, are being developed for real-time, in situ detection of pathogens or toxins. These tools are promising for rapid diagnostics and environmental monitoring. In conclusion, molecular approaches have transformed diagnostic science and strain identification by enabling high speed, high accuracy, and high resolution detection across a wide range of biological systems. Their integration with bioinformatics, artificial intelligence, and portable platforms is shaping the future of personalized medicine, precision agriculture, and disease surveillance. Previous year CSIRNet questions with answers. CSIRNet June 2018. Which one of the following genes is typically used for molecular barcoding of bacterial strains? A18SRRNA. B16SRRNA CCOIDX. Answer: B16SRRNA. CSIRNet Deck 2021. A researcher uses PCR to amplify a pathogen-specific gene from blood samples. Which of the following techniques will best help quantify the amount of pathogen in the sample? A conventional PCR. B.Blot CQPCR, real-time PCR, DRFLP. Answer: CQPCR, real-time PCR. CSIRNet June 2014. In lamp loop-mediated isothermal amplification, which of the following is true? A requires thermal cycling. B uses strand-displacing DNA polymerase. C needs only 2 primers. D only used for DNA sequencing. Answer: B uses strand-displacing DNA polymerase. Let me know your next topic. I'll continue preparing your CSIRNet JRF Life Sciences Applied Biology lectures with the same depth and focus. You said. Genomics and its application to health. ChatGPT said. Here is your CSIRNet JRF Life Sciences lecture on Applied Biology, Genomics and its application to health. Maximum word limit. JRF level. Previous year questions with answers included. No tables. Full paragraph format. Genomics refers to the comprehensive study of the entire genome of organisms, including the structure, function, evolution, mapping, and editing of DNA. It goes beyond studying individual genes to understanding how the whole genome contributes to the functioning of cells and organisms. The emergence of high-throughput sequencing technologies and bioinformatics tools has revolutionized genomics, enabling the decoding of complete genomes across all domains of life. The field has direct and transformative applications in human health, providing insights into disease mechanisms, diagnosis, prevention, therapy, and the personalization of medical care. The Human Genome Project (HGP), completed in 2003, was a landmark achievement in genomics. It provided the first complete sequence of the human genome, identifying about 25,000 protein-coding genes in vast regions of regulatory and non-coding DNA. This has laid the foundation for medical genomics, where researchers now focus on identifying disease-associated genes, understanding gene-environment interactions, and developing targeted interventions. One of the core contributions of genomics to health is the identification of genetic mutations and polymorphisms associated with inherited and complex diseases. Mutations in genes such as BRCA1 and BRCA2 are linked to hereditary breast and ovarian cancers. Similarly, mutations in CFTR cause cystic fibrosis, while HBV gene mutations cause beta thalassemia and sickle cell anemia. Genome-wide association studies (GWAS) have been instrumental in identifying single nucleotide polymorphisms (SNPs) that are statistically associated with complex disorders such as diabetes, hypertension, asthma, schizophrenia, and autoimmune diseases. These findings help in risk prediction, early diagnosis, and patient stratification. Pharmacogenomics, a branch of genomics, studies how genetic variation affects an individual's response to drugs. This allows for the tailoring of medications based on a person's genetic profile, a concept known as personalized or precision medicine. For example, variations in genes like CYP2D6 and 3 cypric-pounds A4 influence how patients metabolize drugs like tamoxifen, warfarin, and antidepressants. Understanding these differences can reduce adverse drug reactions and increase therapeutic efficacy. Genomics has profoundly impacted cancer diagnostics and therapeutics. Tumor genomes often harbor specific mutations or gene expression signatures that can guide treatment. For example, mutations in the EGFR gene in non-small cell lung carcinoma suggest sensitivity to tyrosine kinase inhibitors. Similarly, HER2 overexpression in breast cancer is targeted with monoclonal antibody therapies like trastuzumab. Liquid biopsy, an emerging genomic tool, allows the detection of circulating tumor DNA (ctDNA) from blood samples, enabling non-invasive cancer detection and monitoring of treatment response. In infectious disease, genomics enables the rapid identification and characterization of pathogens. During outbreaks like COVID-19, genome sequencing of SARS-CoV-2 was crucial for tracking viral variants and developing diagnostics and vaccines. Genomic surveillance allows monitoring of antimicrobial resistance genes, virulence factors, and transmission dynamics of pathogens like mycobacterium tuberculosis, Plasmodium falciparum, and Neisseria gonorrhoeae. Metagenomics, which analyzes all genetic material in a sample, helps detect emerging pathogens and study microbiomes associated with health and disease. Prenatal and neonatal genomics is another critical area. Techniques like non-invasive prenatal testing (NIPT) use cell-free fetal DNA in maternal blood to detect chromosomal abnormalities like trisomy 21, Down syndrome, trisomy 18, and trisomy 13. Postnatal genome sequencing can diagnose rare, undiagnosed genetic disorders in newborns, often guiding life-saving interventions. Gene therapy is a direct application of genomics where faulty genes are replaced, corrected, or silenced to treat diseases. The use of CRISPR-Cas9, a genome editing tool, has made it possible to precisely modify DNA sequences. Clinical trials are ongoing for CRISPR-based therapies for diseases like sickle cell anemia, beta thalassemia, and Leber congenital amaurosis. In vivo and ex vivo strategies are being optimized for safety and efficacy. Genomics also plays a vital role in vaccine development, particularly in the design of recombinant and mRNA vaccines. For instance, the mRNA-based COVID-19 vaccines, Pfizer-BioNTech and Moderna, were developed using genomic information from the SARS-CoV-2 spike protein. Reverse vaccinology, which involves mining pathogen genomes for antigenic proteins, has led to vaccines against Neisseria meningitis and holds promise for others like malaria and HIV. In public health, genomic data informs population screening programs, epidemiological studies, and policymaking. Projects like the 1000 Genomes Project and the All of Us Research Program collect diverse genomic data to understand how genetic diversity impacts health outcomes and to ensure equitable healthcare. However, the application of genomics to health also raises ethical, legal, and social issues such as privacy of genetic data, informed consent, and potential discrimination based on genetic information. These concerns are addressed by frameworks like GINA, Genetic Information Non-Discrimination Act, and guidelines from the Human Genome Organization (HUGO). In summary, genomics is reshaping the landscape of healthcare by facilitating precision diagnostics, individualized therapy, early disease prediction, and advanced vaccine development. Its integration with technologies like artificial intelligence, big data analytics, and mobile health platforms holds immense potential for transforming global health, making medicine more predictive, preventive, and personalized. Previous year CSIR-NET questions with answers. CSIR-NET Deck 2016. Which of the following techniques is most appropriate for identifying genetic variants associated with complex human diseases such as diabetes and asthma? APCR. B-gel electrophoresis C genome-wide association study GWAS ddNA cloning. Answer: C genome-wide association study GWAS. CSIR-NET June 2019. A patient with breast cancer shows HER2 gene amplification. Which of the following therapeutic agents is most appropriate in this case? A. Imatinib. B. Trastuzumab, C. Tamoxifen, D. Paclitaxel. Answer: B. Trastuzumab. CSIR-NET Deck 2022. Which of the following is used as a genome editing tool in gene therapy? A. Southern blotting. B. ELISA. CCRISPR-Cas9 ddNA microarray. Answer: CCRISPR-Cas9. Let me know the next topic from Applied Biology, and I'll prepare it in the same high-quality format. You said, agriculture, ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on Applied Biology, Agriculture. Maximize detail, JRF level, based on previous year question trends, fully paragraph-based, no tables, with 2 to 3 previous year questions and answers included. Agriculture, as a branch of applied biology, integrates principles of plant science, genetics, biotechnology, microbiology, soil biology, and ecology to enhance crop productivity, resistance, and sustainability. The modern approach to agriculture emphasizes understanding biological processes at the molecular and organismal levels to improve crop yields, reduce losses, combat climate challenges, and ensure food security. This area is highly relevant to the CSIR-NET syllabus under applied biology, where focus areas include plant breeding, pest and disease management, biotechnology in agriculture, and sustainable farming practices. The Green Revolution, which began in the 1960s, marked the first major application of biological knowledge in agriculture. It involved the use of high-yielding varieties (HYVs) of crops, chemical fertilizers, and irrigation systems. Seminole varieties of wheat and rice, developed through conventional breeding, drastically increased productivity. However, this also led to environmental issues such as soil degradation, water depletion, and pesticide overuse. Consequently, modern agricultural biology has shifted towards sustainable practices, molecular breeding, and genetically engineered crops to reduce environmental impact and increase resilience to stress. One of the significant breakthroughs in agricultural biology is plant genetic engineering. Genetic modification enables the introduction of desirable traits such as pest resistance, herbicide tolerance, drought resistance, and enhanced nutritional content. The introduction of Bt crops, e.g., Bt cotton, Bt brinjal, where the cry gene from Bacillus thuringiensis is inserted into the plant genome, imparts resistance against Lepidoptera and pests by producing insecticidal proteins that disrupt the gut of the insect. Bt cotton has been widely adopted in India and has led to reduced pesticide use and increased yields. Herbicide-tolerant crops are another innovation, such as Roundup Ready soybean and maize, engineered to tolerate glyphosate. These allow farmers to control weeds without damaging the main crop. However, overuse has raised concerns about herbicide-resistant weed evolution and biodiversity loss. Molecular breeding and marker-assisted selection (MAS) have improved the efficiency of conventional plant breeding. Molecular markers linked to disease resistance or stress tolerance can be used to select for desired traits at the seedling stage, accelerating the breeding process. For instance, resistance genes like Exit21 for bacterial blight in rice and LR34 leaf rust in wheat have been successfully deployed using MAS. CRISPR-Cas9 and other gene editing technologies now allow for precise editing of endogenous plant genes without the introduction of foreign DNA. For example, editing of the sweet gene family in rice has provided resistance to xanthomonas infections. Since these edits may not involve transgenes, they can potentially bypass strict GMO regulations, opening new doors for crop improvement. Soil biology and microbiome management are also key areas in agriculture. Understanding the soil microbial community helps in enhancing nutrient uptake and plant growth. Biofertilizers, including nitrogen-fixing bacteria like Rhizobium, Azotobacter, and Frankia, and phosphate-solubilizing microbes, promote sustainable nutrient cycling. Mycorrhizal fungi, especially Arbuscular mycorrhizae, improve phosphorus uptake and drought tolerance. Biopesticides, derived from natural organisms like Trichoderma, Bovaria, and nuclear polyhydrovirus, are gaining popularity due to their specificity and environmental safety. These are used for managing plant pathogens, nematodes, and insect pests in eco-friendly ways. Plant tissue culture techniques, such as micropropagation, somatic embryogenesis, and anthroculture, are widely used for mass production of disease-free leaf plantlets, conservation of rare species, and generation of homozygous lines in breeding programs. Clonal propagation of crops like banana, sugarcane, and orchid fruit tissue culture has revolutionized horticulture and floriculture industries. Climate-resilient agriculture involves breeding crops tolerant to abiotic stresses such as drought, salinity, flooding, and heat. DREB, HKT, and NHX gene families are known to confer stress tolerance. Transgenic crops with overexpression of such genes are under development and testing. Nutritional improvement through biotechnology is exemplified by golden rice, which contains genes from daffodil and bacteria to produce beta-carotene, a precursor of vitamin A. This biofortification approach is being applied to several staple crops to combat micronutrient deficiencies. Integrated pest management (IPM) combines biological, cultural, physical, and chemical tools to minimize pest damage. It includes crop rotation, natural predator conservation, resistant cultivars, and judicious pesticide use. IPM reduces environmental impact and delays pest resistance development. Sustainable agriculture emphasizes conservation of biodiversity, minimal chemical input, and ecological balance. Practices include organic farming, use of compost and green manure, crop diversification, and agroforestry. In conclusion, applied biology in agriculture has evolved from traditional selection to precision genome editing and ecosystem-based management. The convergence of molecular tools, bioinformatics, and ecological knowledge is redefining crop improvement and agricultural sustainability. Understanding these concepts at both fundamental and applied levels is crucial for CSIR-NET aspirants aiming to master this area and solve real-world problems in agriculture and food security. Previous year CSIR-NET questions with answers. CSIR-NET June 2017. Bt cotton plants are genetically modified to express which of the following to resist insect attack? A EPSPS enzyme. B-cry protein gene from Bacillus thuringiensis C glyphosate resistance gene D virus coat protein gene. Answer: B-cry protein gene from Bacillus thuringiensis. CSIR-NET Deck 2021. Golden rice is genetically engineered to be rich in iron. B zinc. C vitamin A, beta carotene, D vitamin D. Answer: C vitamin A, beta carotene. CSIR-NET June 2018. Marker-assisted selection is best described as a hybridization-based crop improvement. B selection based on yield alone. C using molecular markers linked to desired traits for breeding. D physical selection based on plant morphology. Answer: C using molecular markers linked to desired traits for breeding. Let me know the next topic you want from applied biology or any other CSIR-NET unit. I'll prepare it in the same format. You said. Genomics and its application to agriculture. ChatGPT said. Here is your CSIRNet JRF Life Sciences lecture on genomics and its application to agriculture. Maximize word limit. Exam-oriented. JRF level. No tables. No wasted space. With 2 to 3 previous year questions and answers. Genomics, the study of an organism's complete set of DNA including all its genes, has revolutionized modern agriculture by providing precise tools for crop improvement, stress resistance, and yield enhancement. In agricultural biology, genomics is not merely a tool for gene identification but forms the foundation for molecular breeding, transgenic development, disease diagnostics, and sustainable farming practices. The advent of high-throughput sequencing technologies, bioinformatics, and functional genomics has allowed scientists to explore gene function, expression profiles, and regulatory networks in plants at an unprecedented scale. The primary goal of agricultural genomics is to understand how genes control agronomically important traits such as drought resistance, flowering time, disease resistance, nutrient use efficiency, and grain quality. Through whole genome sequencing, WGS genomes of important crops like rice, Oryza sativa, maize, Z maize, wheat, triticum estivum, and tomato, Solanum lycopersicum have been fully sequenced. These reference genomes allow for the identification of genes and QTLs, quantitative trait loci, associated with complex traits. One of the earliest milestones in agricultural genomics was the International Rice Genome Sequencing Project (IRGSP), which provided a complete and annotated rice genome, enabling researchers to link gene functions with stress responses and productivity. Similarly, genomic information in maize and wheat has facilitated the mapping of genes involved in kernel development, disease resistance, and abiotic stress tolerance. Comparative genomics between model plants like Arabidopsis thaliana and crops allows identification of conserved genes and regulatory pathways. For example, the flowering locust TFT gene, first studied in Arabidopsis, plays a central role in flowering regulation and has been targeted in crops to alter flowering time and photoperiod sensitivity. Functional genomics tools such as transcriptomics, RNA-seq proteomics, and metabolomics provide insight into gene expression patterns under stress conditions or developmental stages. These datasets help identify candidate genes that can be used for trait improvement via either marker-assisted selection, MAS, or transgenic methods. RNA-seq data have helped reveal how rice and wheat respond to the gene level to drought, salinity, and pathogen infection. Molecular markers, including SSRs, simple sequence repeats, SNPs, single nucleotide polymorphisms, and AFLPs, are derived from genomic information and used extensively in marker-assisted breeding. For example, SNP markers linked to the sub1 gene in rice, which confers submergence tolerance, have been used in breeding flood-tolerant varieties like Swarna sub1. This allows integration of traits without affecting the rest of the genome, making breeding faster and more precise. Genome-wide association studies (GWAS) use natural population diversity to associate specific genetic loci with traits, offering higher resolution than traditional QTL mapping. GWAS has been used to identify genes related to nutrient uptake, such as nitrogen and phosphorus efficiency, pest resistance, e.g., against stem borers and flowering time in many crops. A transformative development in genomics is the use of CRISPR SARS9 genome editing in agriculture. Unlike traditional GMOs that introduce foreign DNA, CRISPR allows targeted mutation or replacement of endogenous genes. In rice, CRISPR has been used to knock out the BADH2 gene, enhancing aroma. In tomatoes, editing RIN genes has improved shelf life. In maize, Argos 8 gene editing conferred drought tolerance. These gene edits are often indistinguishable from natural mutations and are being explored for regulatory approval as non-GMO. Pangenomics, which involves sequencing multiple genotypes of a species, provides insight into genetic diversity, presence-absence variation, and rare alleles linked to traits. In crops like maize, where genome size and complexity are high, pangenome studies help in utilizing diverse germplasm for breeding resilient varieties. Transcriptome profiling during abiotic and biotic stress helps identify stress-responsive genes. For example, under drought stress, genes encoding late embryogenesis abundant, leak proteins, dehydrins, and DREB transcription factors are highly expressed. These stress-related genes can be used to develop transgenic or gene-edited crops. In pathogen resistance, genomics has enabled the identification of resistance R genes and defense signaling pathways. Resistance genes like Pi54 in rice, against blast disease, RPG1 in barley, against stem rust, and year 15 in wheat yellow rust have been characterized and used in breeding. Genomics-based diagnostics can also detect emerging pathogen strains and help track resistance breakdown. Genomic selection (GS) is a breeding approach where genome-wide markers are used to predict the breeding value of individuals. GS is especially useful for polygenic traits like yield and climate resilience. It reduces the breeding cycle and increases selection accuracy. Metagenomics, which analyzes DNA from environmental samples like soil, helps understand the plant microbiome and its role in nutrient uptake, disease suppression, and growth promotion. This knowledge is being used to develop microbial inoculants and biofertilizers. In precision agriculture, genomic tools assist in tailoring crop management practices to the genetic potential of the variety in the local environment. By integrating genomics with phenomics, high-throughput phenotyping (AI), and remote sensing, farmers can make informed decisions about planting, irrigation, and fertilization. Genomics-assisted biofortification targets genes involved in micronutrient accumulation. In rice and wheat, genes controlling iron, zinc, and vitamin A synthesis have been modified to combat malnutrition. In conclusion, genomics has emerged as the backbone of modern agricultural biology. Its integration into breeding, diagnostics, and crop improvement strategies enables a sustainable, productive, and resilient agricultural system. For CSIRNet aspirants, understanding the principles, tools, and real-world applications of genomics in agriculture is essential, as this area is frequently targeted in the exam and forms the basis for cutting-edge research in life sciences. Previous year CSIRNet questions with answers. CSIRNet June 2019. The sub1 gene introduced into rice through marker-assisted backcrossing confers tolerance to a drought. B salinity C submergence D cold stress answer C submergence. CSIRNet Deck 2020. In crop genomics, the CRISPR SARS9 technique is used primarily for increasing genome size. B inserting random mutations C precise editing of specific genes D generating RNA interference constructs answer C precise editing of specific genes. CSIRNet June 2022. Which of the following statements about genome-wide association studies GWAS is correct? A GWAS only uses SSR markers. BGWAS can identify loci associated with complex traits. CGWAS is less accurate than QTL mapping. DGWAS can only be used in animals. Answer. BGWAS can identify loci associated with complex traits. Let me know the next topic you want from applied biology or another unit of CSIRNet Life Sciences, and I'll continue in the same high detail, exam-oriented format. You said, gene therapy. ChatGPT said. Here is your CSIRNet JRF Life Sciences lecture on gene therapy. Maximize word limit. JRF level. Paragraph format. No tables. Based on previous year questions with answers and year mentioned. Gene therapy is a transformative approach in molecular medicine aimed at treating or preventing diseases by directly modifying the genetic material within a patient's cells. It represents one of the most advanced applications of molecular biology and biotechnology in clinical practice. In gene therapy, defective or missing genes are corrected, replaced, or silenced using various delivery methods. The goal is to address the root cause of genetic disorders, rather than merely alleviating symptoms. The concept of gene therapy rests on the central dogma of molecular biology, where a functional gene produces a functional RNA and protein. In many genetic disorders, a mutation disrupts this flow, leading to a defective or missing protein. Gene therapy seeks to restore this pathway by introducing a correct version of the gene or modifying the existing genetic sequence using tools like CRISPR, SARS9, zinc finger nucleases, ZFNs, or TALENs. These molecular scissors can precisely target and edit specific DNA sequences in vivo or ex vivo. Gene therapy can be classified into somatic gene therapy and germline gene therapy. In somatic gene therapy, the genetic alteration is restricted to somatic cells and is not heritable. It is the only form approved for clinical application in humans. Germline therapy, which involves modification of gametes or embryos, remains ethically controversial and is currently prohibited in most countries due to the risk of permanent heritable changes. Another classification distinguishes in vivo gene therapy, where the gene is delivered directly into the patient's body, and ex vivo gene therapy, where cells are genetically modified outside the body and then transplanted back. Ex vivo methods are commonly used in blood disorders like severe combined immunodeficiency (SCID) and beta thalassemia, as they allow precise control over gene delivery and screening of modified cells before reintroduction. Vectors are essential for gene delivery. Viral vectors such as retroviruses, lentiviruses, adenoviruses, and adeno-associated viruses (AAVs) are commonly used because of their high efficiency in gene transfer. Retroviruses integrate their genome into the host DNA, providing long-term expression but carry risks of insertional mutagenesis. AAVs are favored for their low immunogenicity and stable gene expression. Non-viral vectors, including liposomes, naked DNA, and nanoparticles, offer safer alternatives but usually have lower efficiency. A landmark in gene therapy was the successful treatment of X-linked SCID using a retroviral vector to introduce the IL-2RG gene into bone marrow cells. Despite initial success, some patients developed leukemia due to insertional activation of oncogenes, highlighting the need for safer vectors and precise integration control. The lentiviral vector system, with a safer integration profile, later emerged as a preferred alternative. The emergence of CRISPR-Cas9 technology has revolutionized gene therapy by allowing site-specific editing of the genome with unparalleled accuracy. This system uses a guide RNA to target a specific DNA sequence in a Cas9 nuclease to induce a double-stranded break. The break is then repaired by the cell's endogenous DNA repair machinery, leading to gene correction, disruption, or insertion. CRISPR is being tested in clinical trials for diseases such as sickle cell anemia, Leber congenital amaurosis, and beta thalassemia. In cancer therapy, gene therapy strategies include suicide gene therapy, where a gene encoding a toxic protein is delivered selectively to cancer cells, and immune gene therapy, which enhances the immune system's ability to recognize and attack tumors. One of the most successful examples is CAR T-cell therapy, where T cells are genetically engineered to express chimeric antigen receptors, CARs, that target cancer antigens such as CD19 in leukemia and lymphoma. Gene therapy is also being explored for neurodegenerative diseases, such as Parkinson's disease, spinal muscular atrophy (SMA), and Huntington's disease. For instance, Zolgensma, an AAV-based gene therapy for SMA, delivers a functional copy of the SMN1 gene and has shown remarkable success in infants with this fatal disorder. Despite its promise, gene therapy poses several challenges. These include immune responses to vectors, transient expression, off-target effects, especially in genome editing, and the difficulty of targeting specific tissues. Moreover, large-scale manufacturing, regulatory approval, and cost are significant barriers to widespread clinical use. From a regulatory standpoint, gene therapy trials are closely monitored by agencies like the FDA and EMA. Ethical considerations, particularly around germline editing, human enhancement, and long-term effects, are under active international debate. The first CRISPR baby case in China 2018 triggered widespread condemnation and stricter regulatory oversight worldwide. In agriculture and veterinary medicine, gene therapy techniques are being explored for improving livestock health, productivity, and resistance to disease, though the primary focus remains in human therapeutics for now. In conclusion, gene therapy represents the pinnacle of personalized molecular medicine. Its integration with genomics, bioinformatics, and synthetic biology promises a future where genetic diseases may be not only treatable but curable. For CSIRNet aspirants, mastering the principles, techniques, and clinical examples of gene therapy is essential, as this area is conceptually rich, frequently tested, and at the frontier of biotechnology research. Previous year CSIRNet questions with answers. CSIRNet Deck 2018. Which of the following gene therapy methods is classified as ex vivo? A direct injection of viral vector into muscle B delivery of genes via nanoparticles into lungs. C. Genetic modification of bone marrow cells outside the body and reinfusion. D. CRISPR-Cas9 based gene editing in brain tissue. Answer. C. Genetic modification of bone marrow cells outside the body and reinfusion. CSIRNet June 2021. The risk of insertional mutagenesis is most closely associated with which of the following vectors used in gene therapy? A. Adenovirus. B. Retrovirus. C. Liposome-based vector D. Naked DNA. Answer. B. Retrovirus. CSIRNet Deck 2022. Which of the following statements about CRISPR-Cas9 is correct in the context of gene therapy? It introduces random mutations across the genome. B. It cleaves RNA in a sequence-specific manner. C. It induces site-specific double-stranded DNA breaks. D. It always integrates foreign DNA into the host genome. Answer. C. It induces site-specific double-stranded DNA breaks. Let me know the next topic in applied biology or any other unit of CSIRNet Life Sciences, and I'll continue in the same in-depth and exam-focused manner. You said. Bioresource and uses of biodiversity. ChatGPT said. Here is your CSIRNet JRF Life Sciences lecture on bioresources and uses of biodiversity. Maximize word limit. JRF level. Paragraph format. Based on CSIRNet Life Sciences previous year question areas. No tables. With 2 to 3 solved PYQs. Biodiversity, or biological diversity, encompasses the variety and variability of all forms of life on Earth, including genetic, species, and ecosystem diversity. Bioresources are the components of biodiversity that hold actual or potential utility to humans, such as plants, animals, microbes, and their products. The study and application of bioresources, a core area in applied biology, has profound implications for medicine, agriculture, biotechnology, and sustainable development. The Convention on Biological Diversity (CBD) defines bioresources as genetic resources, organisms or parts thereof, populations, or any other biotic component of ecosystems with actual or potential use or value for humanity. The uses of biodiversity can be broadly categorized into direct, indirect, and option future potential uses. Direct uses involve tangible biological products like food grains, vegetables, fruits, fish fiber, cotton, jute, coir fuel wood, dung medicines, quinine, artemisinin, taxol, and industrial products rubber, resins, oils. Indirect uses relate to ecosystem services such as pollination, nutrient cycling, climate regulation, water purification, and soil formation. Biodiversity also underpins cultural, spiritual, and recreational benefits. Option values reflect the future, as yet unknown, uses of biodiversity, which may be critical for medical or technological innovations. Bioresources in agriculture include crop wild relatives and land processes that carry genes for resistance to pests, diseases, or environmental stress. These are crucial in crop improvement programs via plant breeding or genetic engineering. For example, wild varieties of rice and wheat have contributed genes for drought and salinity tolerance in modern cultivars. Similarly, microbial bioresources are exploited for nitrogen fixation, e.g., rhizobium, azospirillum phosphate solubilization, bacillus, pseudomonas, and biopesticide development, bacillus thuringiensis. In pharmaceutical sciences, over 25% of drugs used in modern medicine are derived from higher plants, many of which are from tropical rainforests. Examples include morphine from Papaver somniferum, aspirin from Salix alba, and vincristine from Catharanthus roseus. Additionally, marine bioresources, such as sponges, corals, and cyanobacteria, are rich sources of novel anti-cancer and anti-inflammatory compounds. Biodiversity also fuels biotechnological innovation. Enzymes from extremophiles like Thermus aquaticus, source of tactile used in PCR, or Pyrococcus furiosus, used in high-temperature industrial processes, illustrate how genetic diversity translates into technological breakthroughs. Metagenomics allows access to genetic resources of uncultivable microbes from diverse environments, enabling novel enzyme and metabolite discovery. Genetic resources are protected under international treaties like the Nagoya Protocol, which governs the access and fair benefit sharing of genetic material. The International Treaty on Plant Genetic Resources for Food and Agriculture (ITPGRFA) ensures farmers' rights and conservation of crop diversity. In India, which is one of the 17 megadiverse countries, bioresource management is governed by the Biological Diversity Act, 2002, implemented through the National Biodiversity Authority (NBA). This act ensures sustainable use, conservation, and equitable benefit sharing. Traditional knowledge associated with biodiversity is documented in the Traditional Knowledge Digital Library (TKDL) to prevent biopiracy, as in the famous case where the U.S. patent on turmeric's wound-healing properties was revoked. A key concern is the erosion of biodiversity, driven by habitat destruction, pollution, invasive species, overexploitation, and climate change. Loss of biodiversity not only reduces ecological resilience but also deprives humanity of potential bioresources, which could otherwise address future health, food, and climate-related challenges. Biodiversity is also being economically evaluated using models like total economic value (TEV), which includes direct use, indirect use, and non-use values, existence, and bequest values. Modern conservation approaches like bioprospecting, searching for valuable genetic and biochemical resources, community-based conservation, and ecosystem services evaluation are integrating science with economics and ethics. In conclusion, bioresources are an integral part of biodiversity with immense actual and potential value. Their responsible utilization, equitable sharing, and long-term conservation are essential for human well-being, sustainable development, and ecological balance. For CSIRNet Life Sciences, this topic is critical because it integrates ecology, biotechnology, policy, and ethics, areas that are often cross-linked in multiple sections of the exam. Previous year CSIRNet questions with answers. CSIRNet June 2016. Which of the following is a correct example of indirect use of biodiversity? A medicinal plants. Bee firewood collection. C. Pollination by bees. D. Collection of wild fruits. Answer. C. Pollination by bees. Explanation. This is an ecosystem service, not a direct harvest of biodiversity. CSIRNet Deck 2019. The Indian law that governs access to biological resources and associated knowledges. Environment Protection Act, 1986. B. Forest Conservation Act, 1980. C. Biological Diversity Act, 2002. D. Wildlife Protection Act, 1972. Answer. C. Biological Diversity Act, 2002. CSIRNet June 2022. Which of the following is a correct statement regarding bioresources? They are restricted to genetically modified organisms. B. They include only plants used for agriculture. C. They include all biotic components with potential utility. D. They are limited to medicinal plants and microbes. Answer. C. They include all biotic components with potential utility. Let me know the next topic in applied biology or any unit from the CSIR Net Life Sciences syllabus. I'll continue preparing JRF level theory notes accordingly. You said, breeding in plants. ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on breeding in plants. Maximum word limit. JRF level. CSIR Net focused. Previous year question based. Paragraph format. No tables. Plant breeding is the art and science of improving the genetic makeup of plants to enhance desirable traits such as yield, quality, disease resistance, and stress tolerance. This discipline, foundational to agriculture and food security, uses principles of genetics, reproductive biology, molecular biology, and biotechnology. Traditional plant breeding relied on phenotypic selection and hybridization, while modern approaches incorporate marker-assisted selection, tissue culture, genetic engineering, and genomic tools. CSIR Net Life Sciences often tests conceptual understanding, breeding strategies, and integration of molecular tools with classical genetics in plant breeding. The process of plant breeding begins with germplasm collection and evaluation. Germplasm refers to the genetic material available for breeding, encompassing long grasses, wild relatives, traditional cultivars, and mutant lines. The variability present in germplasm is the foundation of breeding efforts. Following evaluation for desirable traits, breeders use selection, either mass selection selecting many plants with desired traits, or pure line selection selecting and propagating progeny from a single superior individual to improve populations. Hybridization is the crossing of genetically distinct parents to combine favorable alleles. The resulting F1 hybrids often exhibit heterosis or hybrid vigor, showing superior qualities like higher yield or stress resistance. However, in self-pollinated crops like wheat or rice, hybrid vigor tends to break down in subsequent generations, so selection must follow hybridization to fix desirable traits. For cross-pollinated crops, e.g., maize composite and synthetic varieties are developed through recurrent selection to maintain heterozygosity. Breeding for disease and pest resistance uses gene interactions and relies on the identification of resistance R genes in wild relatives. Classical backcrossing introduces a resistance gene into a high-yielding cultivar while retaining agronomic traits. In the Green Revolution, for instance, IR8 rice was developed through hybridization and selection for dwarf stature and high yield. Mutation breeding, induced by chemicals e.g., AMPs or physical agents e.g., gamma rays creates novel alleles that are then selected for desirable traits. This has been successful in crops like groundnut and barley. However, the mutations are random and require extensive screening. The advent of molecular breeding revolutionized plant breeding. Marker-assisted selection MAS uses molecular markers RFLP, SSR, SNP linked to traits of interest to select desirable plants even in the absence of phenotype. This is particularly useful for traits that are difficult to screen phenotypically, such as root architecture or drought tolerance. The integration of QTL mapping allows breeders to identify genomic regions associated with quantitative traits, essential for complex trait breeding. Genomic selection (GS) is an advanced approach where genome-wide markers predict the breeding value of individuals, increasing selection accuracy and reducing breeding cycles. This is especially relevant in perennials or long gestation crops like coconut or oil palm. Transgenic breeding introduces foreign genes, e.g., BT, herbicide resistance into crops to improve resistance and quality. However, regulatory hurdles and biosafety concerns limit its widespread use. Genome editing, particularly using CRISPR, SOS9, is a frontier in plant breeding. It allows precise edits in endogenous genes to improve traits like flowering time, nutrient efficiency, and stress tolerance without introducing foreign DNA, making the plants non-transgenic in some regulatory contexts. Breeding also addresses abiotic stress resistance, an emerging priority in the face of climate change. Traits like drought tolerance, salt resistance, and temperature resilience are polygenic and complex. The identification of transcription factors, e.g., DREB, NAC, and stress-inducible genes has facilitated molecular breeding for such traits. Plant breeders must also consider intellectual property rights (IPR) and farmers' rights, especially in countries like India under the Protection of Plant Varieties and Farmers' Rights (PPV and FR Act). The balance between innovation and equity is crucial for sustainable breeding systems. In conclusion, plant breeding is a dynamic field integrating classical genetics, molecular tools, and biotechnological innovations. For CSIR Net aspirants, understanding the methodologies, principles, and applications of plant breeding, particularly hybridization, marker-assisted selection, QTL mapping, mutation breeding, and genome editing, is essential. Questions often test your ability to connect genetics with applied breeding outcomes and understand trait inheritance under various breeding schemes. Previous year CSIR Net questions with answers. CSIR Net Deck 2016. Which of the following is an advantage of marker-assisted selection (MAS) in plant breeding? It allows direct visualization of desired traits. B. It eliminates the need for hybridization. C. It allows selection of plants based on molecular markers linked to traits. D. It increases heterozygosity in crops. Answer. C. It allows selection of plants based on molecular markers linked to traits. CSIR Net June 2020. Backcross breeding is generally used to generate heterozygous lines. B. Introduce a single gene into a superior variety. C. Create transgenic plants. D. Develop polyploid varieties. Answer. B. Introduce a single gene into a superior variety. CSIR Net Deck 2017. In marker-assisted selection, the use of molecular markers is advantageous because they are always dominant markers. B. They eliminate the need for any phenotype evaluation. C. They can help in selecting traits not expressed early in development. D. They are only applicable in mutation breeding. Answer. C. They can help in selecting traits not expressed early in development. Let me know the next topic you'd like detailed. We can continue building your full applied biology coverage or move to another unit of the CSIR Net Life Sciences syllabus. You said, and animals, ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on breeding in animals. High quality, maximum word limit, paragraph format, CSIR Net Life Sciences focused, with previous year questions and answers. Animal breeding refers to the controlled propagation of domesticated animals to improve desirable traits such as milk production, meat quality, egg laying capacity, disease resistance, fertility, and growth rate. It forms a critical pillar in animal husbandry and sustainable livestock development. In the context of CSIR Net JRF Life Sciences, questions focus on classical and modern genetic principles applied to animal breeding, inheritance of quantitative traits, artificial selection strategies, and integration of molecular tools like marker-assisted selection and genomic selection. The foundation of animal breeding lies in population genetics and quantitative genetics. Traits of economic importance in animals are usually polygenic, influenced by multiple genes, and show continuous variation, making quantitative trait loci, QTL, mapping, and heritability estimation essential components of animal breeding programs. Selection intensity and response to selection depend on the heritability of traits and the selection differential, as explained by the breeder's equation. R equals h squared times s, where r is the response, h squared is heritability, and s is selection differential. Inbreeding and outbreeding are two principal strategies in classical breeding. Inbreeding involves mating between related individuals to concentrate desirable alleles but increases the risk of inbreeding depression, where deleterious recessive alleles become homozygous. This is sometimes strategically used to fix desirable traits in pure lines, such as in laboratory model organisms like mice. Outbreeding, including crossbreeding, outcrossing, and species hybridization, aims to enhance heterozygosity and exploit heterosis, hybrid vigor, leading to improved performance in F1 individuals. For example, crossbreeding of Bos indicus Indian breeds with Bos taurus exotic breeds has enhanced milk productivity in cattle under Indian climatic conditions. Artificial insemination (AI) is a pivotal tool in modern animal breeding, allowing for genetic dissemination from elite males across large populations, even internationally. It reduces disease transmission and improves selection accuracy. AI forms the backbone of progeny testing, wherein the performance of offspring is evaluated to determine the genetic merit of breeding sires. This is a common practice in dairy cattle improvement programs. Open nucleus breeding systems (ONBS) represent structured breeding strategies where superior animals are maintained in a nucleus herd and genetic gains are disseminated to commercial herds. This hierarchical model helps maintain genetic progress and control in large-scale programs. The integration of molecular markers in animal breeding has revolutionized selection accuracy. Marker-assisted selection MAS uses DNA markers, e.g., SNPs, microsatellites tightly linked to QTLs or major genes. 4.